Topic of discussion of this video is on double fertilization in angiosperms, discovery and events. Hope the content of this video will be useful for both undergraduate and postgraduate students of botany studying in different colleges and universities in Karnataka. Contents of this video: introduction, discovery of double fertilization, events before double fertilization, screening for self-pollen, and competition amongst Homo-Specific pollen, events after pollen landing on stigma, pollen adhesion to stigmatic surface, pollen hydration, germination of pollen grains, pollen tube growth in stylar tissue, with a brief account on Kalos Plugs, wet and dry stigma, solid and hollow styles. Then entry of pollen tube into ovule, discharge of male gametes, Syngamy and triple fusion, events after double fertilization, a brief account on polytubie and polyspermy, as well as on preferential fertilization. Double fertilization is a defining feature of reproductive development in most evolutionarily successful and wonderfully diverse group of plants on Earth known as angiosperms. In the embryo sac, two sexual fusions occur. One is syngamy, that is the fusion of one male gamete with the egg, and another is triple fusion, that is fusion of other male gamete with the polar nuclei or secondary nucleus. And therefore, the phenomenon is known as double fertilization. This phenomenon of double fertilization was discovered by S.G. Nawaschin in 1898 in Lilium and Fritillaria species. As a result of first fertilization, a diploid zygote is formed, which is the mother cell of the embryo. The nucleus of the triple fusion product, that is primary endosperm nucleus, is triploid in nature, and this is the first nucleus of the endosperm. Then discovery of double fertilization. Strassburger in 1884 discovered the mechanism of fertilization, that is the fusion of the male and female gametes resulting in embryo formation in angiosperms. His work was mostly devoted to the nuclear cytology of pollen grains and pollen tubes of Gloxinia, Orchis, and Monotropa. However, understanding of the fate of the second male gamete discharged by the pollen tube and the source of origin of the endosperm remained as a major hurdle in gaining a complete insight into the dynamics of fertilization in angiosperms. Nawaschin in 1898, a Russian botanist, showed that in ovules of Lilium and Fritillaria, both male gametes from the pollen tube penetrated the embryo sac. One of them fused with the nucleus of the egg cell, the other fused with the polar fusion nucleus floating in the central cell, initiating second fertilization event. Guinard in 1899 also confirmed the phenomenon of second fertilization while working on two species of Lilium. Sargent in 1899 re-examined the previously prepared slides of fertilized ovules of fusion of the second sperm with the polar fusion nucleus in Lilium martagon and confirmed the second fertilization event. Coulter and Chamberlain in 1912 devoted a monograph entirely to the topic of fertilization in seed plants in their classic book on the morphology of angiosperms. They describe over 60 species coming under 40 genera and 16 families of angiosperms with second fertilization event. Events before double fertilization. Screening for self-pollen. There are ample opportunities for the pollen of the same plant landing on the stigma. This results in inbreeding depression and also reduces genetic variability and adaptability to changing environment. To overcome this, the flowers of different species have variously adapted to avoid self-pollination, like unisexuality of flowers, self-sterility, dichogamy, heterostyle, hercogamy, etc. Then competition amongst homo-specific pollen. The number of the homo-specific pollen grains landing on the stigma is many times more than the number of ovules available for fertilization. Therefore, not all the homo-specific pollen grains are able to participate in fertilization process. Usually, the most vigorous pollen grains will succeed in fertilizing the female gametes by producing the pollen tubes early, and less vigorous pollen grains fail to get a chance in fertilization process. Then events after pollen landing on stigma. The entire pollination and fertilization process consists of a number of successive steps initiated after pollen landing on the stigma, and they are pollen adhesion to stigmatic surface, pollen hydration, pollen grain germination, pollen tube growth in stellar tissue, pollen tube entry into ovule, discharge of male gametes, Syngamy and triple fusion. First one, pollen adhesion to stigmatic surface. Once the pollen grains lands on stigma's surface, they form protective contacts with the pistil tissues. The mature pollen cell wall includes three main layers with some variation between the different species. The wall layers are pollen coat that fills the empty cavities of the exine, and there are lipids, pigments and proteins within it. And then exine wall, that is outer strata composed of sporopollenin and broken by gaps called apertures. And the third one is intine, that is the internal strata primarily made up of pectin and cellulose. The pollen coat extrudes proteins and lipids on the stigma to form a pollen foot, which is crucial for pollen contact with the dry stigma. The second event, pollen hydration. Pollen grains need to absorb water from the stigma for hydration and germination. During pollen hydration, the stigma functions as a source of water. In Arabidopsis, lipids and proteins from the pollen coat, and stigma plays a pivotal role in pollen hydration. After hydration, the pollen grain transforms from nonpolar to highly polarized and organizes its cytoplasm and cytoskeleton to extend a tubular structure. Then pollen germination. When the pollen is shed from anther, it has usually two cells, generative cell and vegetative cell, which is also called as tube cell. The generative cell forms two male gametes, as you can see in this diagram. After that, the vegetative cell forms a pollen tube. The stigmatic fluid secreted by the stigma contains sugar, lipid, and resins, which provides suitable medium for the germination of pollen grains. The growth of the pollen tube of flowering plants is restricted exclusively at their apical regions. Most of the cytoplasm is restricted to the apical region, while a large vacuole fills the grain and older regions of the tube. The cytoplasm is restricted to the apical region of the growing tube by the formation of series of Kalos plugs at regular intervals behind the tip. And in this diagram, you can see the aggregation of Golgi vesicles, endoplasmic reticulum, lipid particles, starch grains in the cap block region. Then what are Callose plugs? The Callose plugs are formed as a ring on the inner side of the tube wall and gradually grow toward the center, which finally seals off the growing tip from rest of the pollen tube, as you can see in this diagram. The apical region of the tube possesses a clear cap called cap block with more granular elements behind. The cap block disappears with the termination of the growth. Then pollen tube growth in stylar tissue. During pollen tube growth in the pistil, it has to penetrate different pistil tissues. Turgor pressure is supposed to play an important role in this process. In species with wet stigma and solid style, the cuticle of the stigma or papillae is disrupted during the secretion of the exudates. Thus, there is no physical barrier for pollen tube entry into the intercellular spaces of the transmitting tissue of the stigma. In the species with wet stigma and hollow style, pollen tubes grow on the surface of the stigma and enter the stylar canal. In species with dry stigma and solid style, the cuticle provides the physical barrier for the pollen tube entry. The cuticle is eroded at the point of contact by the activity of cutinase released by the pollen. After the digestion of the cuticle, the tube enters the pectocellulosic wall of the papillae and finally grows through the intercellular spaces of the stigma and the style. Types of stigma. There are two types, that is dry and wet. Dry, in which the stigma surface is free from any visible exudates, as in case of the members of Asteraceae and Poaceae. Wet type, in which the surface is covered with exudate of varying quantity, as in case of the members of Leguminaceae, Solanaceae and Orchidaceae. Types of styles. There are two types, namely solid and hollow. In the solid style, transmitting tissue with large intracellular spaces connects the stigma with the ovary. The intracellular spaces contain extracellular components secreted by the cells of the transmitting tissue. In solid styles, pollen tubes grow through the intracellular spaces of the transmitting tissue. In hollow style, a canal bordered by one or few layers of glandular cells called canal cells, which connects the stigma with the ovary. Here also, extracellular components fill up the whole canal or the inner lining of the canal cells. In hollow styles, pollen tubes grow through the stylar canal on the surface of the canal cells. And in this diagram, we can see both the solid styles as well as the hollow style in longitudinal section as well as transverse section. Pollen tube entry into ovule. The pollen tube may enter into the ovule via three routes. Porogamy. Pollen tube enters the ovule through the micropyle. Chalazogamy. Pollen tube enters the ovule through the chalazal end. Mesogamy. Pollen tube enters the ovule through the integument or through the funiculus. In most of the angiosperms, the pollen tube enters through the micropyle. Pollen tube entry into embryo sac. It does not matter through which way the pollen tube enters into the ovule, but it always enters into the embryo sac from micropylar end. The entry may be via different passages, like first one is between the egg cell and one of the synergids, and second one is between the walls of the embryo sac and one of the synergids, and third one is directly penetrates one of the synergids. After the pollen tube enters into the micropyle, it will meet one of two synergid cells. The filiform apparatus of synergid cells plays a critical role in pollen tube reception. Discharge of male gametes. After reaching the embryo sac, the pollen tube bursts at its tip and delivers two male gametes. Just prior to the bursting of pollen tube, the tube nucleus disorganizes. Immediately after releasing, the male gametes show amoeboid movement and one male gamete moves towards the egg and other move towards the polar nuclei. And in this diagram, you can see the fate of synergids before and after the entry of pollen tube. Syngamy and triple fusion. One sperm cell fuses with the haploid egg cell, generating the diploid zygote. The fusion of male and female gametes is known as fertilization. The most significant discovery made by Strassburger in 1884 while working on Monotropa, triple fusion. The second sperm cell fuses with the two polar nuclei or secondary nucleus and so forms triple fusion nucleus called primary endosperm nucleus. As discussed earlier, S.G. Nawaschin in 1898, while working with Phytolleria and Lilium, discovered the phenomenon of double fertilization and triple fusion in angiosperms. Then events after double fertilization or post-fertilization changes. After two fusion events, that is double fertilization, following changes will occur. The zygote develops into the embryo. The primary endosperm cell develops into the endosperm. Ovule develops into the seed. Ovary develops into the fruit. And this completes the sexual reproduction in flowering plants. Now, a brief summary on two important aspects, that is polytuby as well as preferential fertilization. The first one is polytuby and prevention of polyspermy. When two sperm cells are delivered inside the embryo sac, both the sperm cells must function in fertilization. However, plants also possess mechanisms to achieve fertilization if gamete fusion fails. A mechanism known as a fertilization recovery associated with polytuby, that is supernumerary pollen tube formation, which becomes active if a defective sperm cells are delivered by the first pollen tube. In this situation, the secondary pollen tube are attracted by the remaining second synergid cell and deliver the sperm cargo to the female gamete. This mechanism provides plants with an additional means to maximize reproductive success. Now, the second aspect is preferential fertilization, a unique mechanism in gamete recognition that remains to be understood is how one sperm attaches and fuses with the egg cell while the second sperm cell simultaneously fuses with the central cell. With the exception of a few plant species such as Plumbago zeylanica containing dimorphic sperm cells, where the large sperm cell containing more mitochondria fuses preferentially with the central cell and the smaller sperm cell with the egg cell. Sperm cells in most flowering plants are isomorphic, and preferential fusion with either of the female gametes has not been observed. Hence, more studies have to be made to understand the existence of preferential fertilization in angiosperms. Thank you for watching.